Hello and welcome to this video. In this video, I'm going to demonstrate the Vollseg Napari extension plugin called Vollseg Napari Trackmate. Now, Vollseg Napari Trackmate is a plugin which is used to visualize and do some track analysis from the tracks that come out of the Fiji plugin Trackmate. Trackmate is a popular Fiji plugin which can take in as an input 3D plus time hyperstacks of raw and segmented labels, where the segmented images can come from any algorithm, for example from Vollseg. Once you input the hyperstack into Fiji, you can use integer label detector to do the tracking. And once you have done the tracking, you can use our automated algorithm called trackmate oneat to automatically correct the mitotic and apoptotic cell trajectories. And not only that, Trackmate also comes with a track scheme where you can manually view your tracks and also correct the tracks if any needed. And once you have done the tracking in Trackmate, you get as an output the XML and the CSV file of the tracks. And now in this plugin, we have made a possibility that you can import those XML and CSV file from Trackmate into Napari to visualize the tracks that you have created in Trackmate in Napari. And not just visualize them, you can visualize your tracks with different track attributes, you can color your tracks with different properties, you can also do some analysis of your tracks in the plugin itself. And most importantly, in this plugin, we convert the segmented images into point clouds using an autoencoder model, which is trained to convert 3D segmented labels into 3D point clouds so that we can compute the properties, the shape features of your cells in the tracks more accurately than is possible using the classical segmentation approaches. So let's dive into it. So documentation of the plugin is on the Napari Hub. The plugin itself can be downloaded from Napari Hub. And just to start using it, I would recommend that you use the samples that we have provided in which we have done the tracking on a hyperstack. So if you go to open sample and click on test track data, and if you're doing this for the first time, the tracking package, the tracked package is going to download from Zenodo by itself. And what it will show you is this hyperstack image, which contains the segmentation one channel and the raw image in the other channel. So you can see over here, this is a 3D plus time plus channel data set. And along with this image, you also will get the XML and the CSV file that we have obtained after doing the tracking from Trackmate. So first thing that I'm going to do is I'm going to actually split this hyperstack using a simple script into labels and raw image separately. So you can delete this hyperstack and instead you can use this label overlaid on the raw image as an input to the plugin. And then you can start the plugin from plugin Napa Trackmater and it will show you the plugin menu on the right hand side. Now you see that there are a lot of options over here and I'm going to quickly fill up these options for you. So let me just grab this window where I have already done this. Here I have already filled up these fields that you have to choose the input image. Optionally, you have to choose the label image, which is the segmentation image that you get from the sample. Then you can select the XML file of Trackmate and the CSV files of track spots and edges that come out of Trackmate. And right now, you see that there are a lot of empty fields because in this particular mode, we are just taking the outputs that come from Trackmate. We hit this orange button and it's going to extract the properties or all the tracks from these XML file, and then it's going to fill up all the fields that you need for actually visualizing the tracks over here. And this is the vanilla analysis of Trackmate. Let's say you don't have any autoencoder models, you just want to take your tracks from Trackmate, but now you want to visualize those tracks in Napari using the 3D viewer. That's all you have to do. Take your XMLs, take your CSV and click on the orange button, and then you get all the tracks. And not just all the tracks, you get some statistical plots out of them. For example, here you can see I have some statistics on the number of spots, gaps, or the starting point, the displacement of the tracks. You have some statistics about the mean speed of your tracks, and all of this is for dividing tracks because that's by default selected. Now if I select on non-dividing tracks, what I'm going to see is my plots are going to get updated because now I'm seeing the statistics coming for the non-dividing trajectories. So in this way, you can nicely separate the analysis between dividing and non-dividing trajectories, but not just by looking at the histograms. You can also look at the temporal statistics. So in this particular plot, you are answering the question that if you have all the tracks at all the time points, and now I want to ask the question that at a given time point, what was the mean value and the variance of the cell to tissue distance for this particular track? Or it could be, I want to know the instantaneous directional change for all of the tracks, the mean value and the variance. So these are the mean and the variance plots for the dynamic features and also for the shape features. And you can just select from non-dividing, you can go back to dividing, and even these plots are going to automatically update, and you will get just that feedback from this plugin. And then if you look at the phenotype analysis, if you haven't selected any of the tracks so far, it's going to just show you the cell intensity of all the dividing tracks and its Fourier transform, so you can see if there's any oscillation in the intensity or not. But now if you select from this drop-down menu one particular track, let's say track ID 8, you are going to see much more analysis plots of this particular track. So you're going to see that this is the speed of the mother and the daughter cells because we are analyzing the dividing cells. You will see the motion angle, radial angle, acceleration, and all of these different dynamic and statistical and shape properties are actually explained in the documentation of what we actually mean by them. And now you can also go to the menu and the tracks also appear over here. So the label images I can turn off, and then I can just look at my track, which is this one. So you can see the cell and you can see the track, and it's by default colored by the track ID. So you can go to the left panel and you can see I want to color this with radius. So it will be colored by radius and you will see your track colored by radius. Or you can say, actually, I want to color my tracks with the generation ID. And so the generation ID means that the mother cell becomes purple and then the daughter cells acquire their own color. And at any time you can change the track. This was track ID 8, you can go to track ID 1, and then it will, the plugin is going to automatically update, showing you the tracks layer for that selected track and updating the plot for that particular track. And by default, it's going to put the color of the track ID, which you can change from the menu on the left. And here I always like seeing this generation tracks. So you can see purple is the mother, then it becomes green, and if it splits again, it changes color and the two daughter cell acquires the same color. You can adjust the tail length, and that's just coming from the napari, core napari feature itself, that you can actually play with these tracks just from the panel. Right. And then you also have the table in which you have the track ID, frame position information of your tracks as a table, which you can save as a CSV file. And then you have this feature where you can color your tracks with any spot or track attributes, and not color the tracks, color the segmentation image. Because as you see here, this is the label image which was input, which was the segmentation results that we got from Vollseg. But now I say I want to actually just color it with track ID. So I select track ID, I hit run, and then it's going to produce another layer of labels where the color of the cells now represents the track ID to which they belong to. So we just give it a minute, and then it should put another layer on the left hand side. And that we see over here. So now if I turn off the labels layer, I see, now you can see if a cell is in a track, it always has the same color, right? And you can use any of these attributes to color the cells in the track, not just color the track itself. And another feature is if, for example, I do a double click at any location where there's a segmentation label, it's actually going to show me the track for that particular cell. So now let's see what we get here. So we clicked, and maybe there was just a very short track that you can see here in purple. So I can just see that in radius, and that's the track that you see over here. And you can ask for the track ID by just showing the ID, and it will display this ID over here. And maybe it's not clear. So maybe you can see the ID much more clearly. So now that's the vanilla analysis that you get from TrackMate, and we have just visualized and kind of analyzed this in Napari, but there's more to this plugin. Now you saw here in the beginning that I had some empty fields, like what is this master XML file and what are these autoencoder models. Now that's the thing, especially in 3D data, that quantification of shapes in 3D data just from the segmentation labels is kind of inaccurate. You can measure the radius by assuming that it's a sphere, or you can just compute a bounding box on it, or you can somehow approximate the surface area by mashing cubes. But it's more nicer to actually use a trained autoencoder model to convert these segmentation labels into point clouds and then compute the properties, the shape features from that autoencoder model. And here you have the possibility of either using a pre-trained autoencoder model, and what it's going to do is it's going to apply that autoencoder model on all of the cells in the tracks for which the segmentation label is present, and convert it to a point cloud, recompute the shape features and the dynamic features, and it's then going to give you a master XML file which contains these updated attributes which are not present in the trackmate XML file. So as I said, you can actually do this in the plugin using a pre-trained model or a custom autoencoder model, or you can use a script, because when the script is running, it's running over all time points and it's a GPU intensive job. So for this, you don't really need Napari. So we have made a script which can utilize multiple GPUs, and it's based upon Lightning. And using multiple GPUs, you can get the prediction of a trained autoencoder model on your label images, and then it will generate a master XML file. So instead of doing analysis with trackmate XML file, now I can just go and do my analysis with the master XML file, and I have to run the compute button again, so it recomputes the properties based upon this master XML file, and then it will update those plots that you saw, and you can again visualize, for example, particular track, let's say track ID1, and now you can also see the surface area or eccentricity kind of features that you have computed using this autoencoder model in the color of the tracks. So for example, now I can say I want to see how was the surface area, and this will color the tracks with the surface area. And you can also click on all tracks, and then it will show you all of those tracks. All right, now comes the most important point. You have done here some visualization, right? You have seen the tracks, you can color them with different features, attributes. You can obtain the segmentation images, the labels with different trackmate features. But most important point of doing this is actually to get some physics out of it, or biological truths out of this. So once you have done this, you have created this master XML file, you can combine this master XML file with these CSV files to convert each of the track into a 2D matrix, where the columns of the matrix are the shape and the feature vectors that we have computed: the radius, volume, surface area, centricity, distance of the cell from the tissue, the relative angle of the cell to the tissue, and so on. All of those dynamical and radial features appear as columns of that 2D matrix, and the rows contain the time. So in the script that comes after this plugin, which takes as an input this XML and the master XML and the CSV file, it converts that into each of the track into a 2D matrix. And once you have that in a 2D matrix, you can use you can do PCA on them. You can use k nearest neighbors, as we do in our script, to classify your tracks into different classes. And once you have that, it will generate a cluster label CSV file that you can input in this plugin. So let's just do that. Let me actually just quickly give it the path so it knows where to go. And I'm going to use the PCA cluster dynamic. Yes, so I've done PCA on these 2D matrices, I've clustered them using the dynamic features. You can choose either to use dynamic features or shape features or both. And then you can open them here. And once you have done this, you hit the compute button again. It's going to now give you another tracks layer where the tracks by default are colored by track ID, but they are also having a property that can color them with cluster ID. So here we are trying to classify. So for example, in this one, we are trying to classify the tracks into cluster classes. So it's not classifying each cell, it's classifying each track into a given class. So a whole track or a tracklet is going to actually have a single color which tells you the class to which it belongs to, and this class is computed using machine learning algorithms. And then you have a whole workflow that is complete. You use Vollseg napari to get the segmentation of your 3D cells. You use autoencoder model to get the point cloud representation. You use that to compute all the or update all the features of the cells in your tracks. You can visualize them and you can cluster them into classes, and now you have a wholesome analysis of your tracks, which is covered with a lot of deep learning approaches, and so is very accurate if you have trained the models on your own data. And now you are close to answering the biological question of your interest on how these cells decide their fate, for example, or any other kind of question that you may have, that do the cells start in the center of the tissue and then move to the border, or how is the dynamics happening in my particular experiment. And that's where I leave you to it, to use the plugin, do the tracking, and if you want our inputs, we are always happy to help you in creating these tracking data that actually helps you then get the science out of your experiment. Thank you for listening.